So it's really the identification of all the different cell types in the mouse colonic tissue. So there are some data processing steps. The first one is filtering based on the QC plot, and I'll talk a little bit more about that and how I decide what to filter. Then it comes to the identification or the separation of colonocytes versus the non-colonocytes. These non-colonocytes are really just going to be the immune and the stromal cells that are also in these samples. And then once I can separate those, I can focus on the colonocytes, and I can then start to try to identify all the different subtypes of colonocytes in these crypts. And I'll talk again more about this in a bit. So this is the colon tissue. So we isolate this colon tissue. This is a human, but we get it from mice. And we have now started using the whole intact tissue. And what I mean by that is before we were just kind of taking off this top part, this epithelial layer, and then sequencing that. But we do know that down here is in the muscle layer is where all the blood flow is, all the immune cells are, and they are also responding to things like diet. So we're now interested in looking at the whole colon tissue. So we now have all tissue there. This is kind of what it looks like at a cross section. At the top here, you have the epithelial cells. These are the colonic crypts that are right here. And then down here, this is where you have your immune cells, your blood vessel cells, muscles, all different kinds of fibroblasts that give nutrients to the colonic crypt stem cells at the bottom. It's all there. So everything is important, and I think it's good that we have started doing this, but it does make the data a little bit more challenging. And as you can see within these crypts, there are a ton of cells really squeezed in here. And even in the center of the crypt, some of these circular things are part of goblet cells. So if we use a different staining, we can kind of see that the blue here are those goblet cells. And so we can capture all of this with our single cell data. So we have all of these different cell types here in the colon. This is just a simplified version of that. And this is what we've been using to do some RNA-seq. So the first thing, like I mentioned, is the QC filtering. This is really going to depend on whether you're using Flex or the 3' sequencing. The Flex is really clean data. You almost don't need to filter this data. If you do filter it, it would be just to kind of get rid of these cells that have a high mitochondrial percentage. But everything else here, to me, kind of looks fine. But if we come over here and we look at the 3' expression data, if we just look at the number of genes, this graph here, just from my experience, I've learned that down here in this section is really kind of junk cells. So we want to kind of filter this out to where it looks a little bit more like this over here. Even in the number of reads here, where there's this break right here, below that is kind of just these junk cells that, again, that we need to filter out before we begin processing the data. And that's how I filter my data. I have to look at the QC plots and kind of just go from there. Again, the Flex data that I'll be showing today is pretty clean, so I actually didn't perform any filtering on this data. This data comes from notobiotic mice, that is mice that don't have a microbiome. So this is pretty much just pure colon tissue, unaffected by the microbiome. This is what the data looks like when we load it into Dr. Sai's software, and then we can begin to embed it. I like using the UMAP because it just makes a lot more sense to me biologically compared to the other embedding methods that you can use. So the first thing when we have this plot, like I mentioned, is going to be trying to separate those colonocytes from the non-colonocytes. I use two major markers for this. I use EpCAM and ECadherin. These are going to be expressed highly in epithelial cells. And this is what I use for me to separate the colonocytes, which are EpCAM positive, from the non-colonocyte cells over here. Of course, this isn't enough. I have to go in and really dig deep into what these cell types are. So just so that you guys know, about 60%, it's really about 50 to 66% are colonocytes. And the other percentage, 34% is going to be these non-colonocytes. And again, this is going to vary. These are mice that don't have a microbiome. So in the normal wild type mice that we do see, this is going to change a little bit. There might be more immune cells because of the microbiome. I'll just really briefly talk about the non-colonocytes, but it won't be my main focus because these cells are actually pretty easy to identify. You can see that they make up just these little clusters here, and they separate really nicely, and we can identify them. And we get all different sorts of immune cells, B cells, macrophages. Sometimes I see dendritic cells, sometimes not. There's also blood vessel cells, so endothelial cells, smooth muscle cells, mural cells, and pericytes that are all connected to the blood vessels. Then there's all sorts of fibroblasts, some myofibroblasts, and then some other types of fibroblasts that like I mentioned, are providing growth factors and nutrients to the colonic stem cells. And then we also see things like neurons and even adipocytes show up in this data. This is just a heat map for all of these cell types, so they do kind of break out really nicely. And I'm pretty confident in these cell types too, but I haven't put a lot of focus into this. I haven't really analyzed data coming from the non-colonocytes just yet, because my major focus has been on the colonocytes. So this is when I subsetted out the colonocytes, and then I begin to try to cluster these cells and see what are these cell types. So I use k-means clustering, and I kind of just play around with the numbers until I find one that really separates out everything kind of the way I like it, like visually, and then I can kind of go from there. So the first thing I do with the colonic cell types is going to be separating out the enteroendocrine cells and the tough cells, and that's just because they actually stick out from the data. So in this case here, this cluster 11, I know is going to be the enteroendocrine cells, and this cluster over here, number 3, is going to be the tough cells. And I know that by the marker genes that they're expressing as well, and we can actually calculate a score for these marker genes for the enteroendocrine cells. So you can see here they scored really highly compared to the rest of the clusters. That's cluster 11 up here, so I know those are enteroendocrine cells. And the tough cells, again, scored really highly and different from the rest of the cells. That's number 3 right here. So these guys are tough cells. That's easy. That's actually pretty easy and shouldn't be too much.
It's hard to me, but
like again, it's just because they actually pull away from the rest of the cells, because they are a differentiated cell type, but they are technically a specialized cell type as well.
Expression is so different; they just separate from the rest.
Exactly. The other cell types that I can try to pull out are goblet cells. Goblet cells are going to be this whole chunk right here. And I know that based on the MUC2 expression, so you can see that it expressed very highly in these clusters here. So I know those are goblet cells. I can also calculate a score for them, and they do separate out compared to the rest of the clusters down here. So I know, again, these are goblet cells right here. So I can actually subset out the goblet cells again, and then begin to focus on the specific goblet cell subtypes. I know nobody really does this, but I thought it was very interesting because once I saw this, I remembered a paper that I had read that actually mentioned that there's 2 lineages from the goblet cells. There's a canonical and a non-canonical lineage, and that's actually what I observed here. So that was pretty cool. Down here, what I'm showing is MKI67. This is a proliferation marker. So you can see that the clusters about right here are going to be the cells that are proliferating.
so that's interesting because they're supposed to be generally quiescent, right?
Yes and no.
So there are actually some cycling cells in there.
Yeah.
They're going to divide. Interesting.
There's also differentiation markers. So for goblet cells, a good differentiation marker is MXD1. And you can kind of see that, I didn't do a good job of lining these up right, but down here are going to be where those proliferating goblet cells are. And actually this little cluster right here that you see is secretory progenitor cells, or what I think are secretory progenitor cells. And then we have the non-canonical goblet cells here using these 2 markers that I grabbed from that paper. So this kind of branch right here is non-canonical goblet cells, and these over here are canonical. You can kind of see that this non-canonical branch is actually very close. That's this branch right here. It's very close to the other colonocytes right here. So it was very interesting to actually see that out on this 3D map. But based on those markers,
that's a biology.
Yeah.
That's right.
To us, just 2 branches. We don't know.
Yeah, I know, and I remember that
complicated.
paper. So for the secretory progenitor cells, they have different names and people call them different things. That's what makes this also very complicated. So I started calling them secretory progenitor cells. I believe previously we were calling them deep crypt secretory cells. So they do have this Reg4 marker. You can see that cluster number 12 is scoring really high for this Reg4 marker. That's this cluster right here. And you can also see that clusters 10 and 12 are also proliferating. This is a proliferation marker, so I know these are proliferating right here. DLL1 and DLL4 are expressed again in these secretory progenitors, and you can see that cluster number 12 is scoring high. It's actually the only one that's kind of scoring. So to me, these are secretory progenitor cells. This is what it looks like once you actually apply those cell types. So we have the secretory progenitor cells here. We have some proliferating goblet cells here, and then the rest, even though there's two lineages, I merged them just to simplify things. Those are the differentiated goblet cells. And actually, if we look at the differentiation potency, you can see that the secretory progenitor cells score really high, and the proliferating goblet cells, and then the differentiated goblet cells at the bottom. So this looks good. That's what I expect from this data. Like I mentioned, we have these differentiated cells, but they're also very specialized cells. They have nice functions. And so you just saw that I was able to identify all these cell types here. The next thing would be to identify the stem cells and the absorptive colonocytes. So you can kind of see from this image to this image, I got rid of all those specialized cells. And now we're really left with the backbone of what I guess colonocytes do. So the colon is largely responsible for absorbing any sort of water that the small intestine didn't absorb. So that's what these cells, that's their function, and that's why they're called absorptive colonocytes. And so what I did was I subsetted out the stem cells and the absorptive colonocytes. They form kind of like this gradient, very similar, I guess, to this crypt right here. They form this gradient from stem cells and transient amplifying cells down here, all the way up to differentiated cells over here. So I know that because of LGR5 expression is located down here at these clusters at the bottom. You can see that these are also the same cells that are proliferating based on proliferation markers. And then there's some differentiation markers that I use. I like to use KRT20 to identify the absorptive colonocytes. And aquaporin 8, this is, of course, a receptor that is responsible for pulling in water into the cells. And that is what I use to identify the late absorptive colonocytes. I don't believe I actually have an image of that, but this is what I'm currently working on right now. This is what I've had to do to be able to identify these cell clusters. My initial thinking when I first started all this single cell stuff was that, we'd be able to have our cell atlas with all our gene markers and cell types and just kind of, click and play and it'll identify stuff for us. I learned over a year or two that is not possible at the time, especially not for this particular cell type. It works actually pretty well for things that separate out really nicely, like blood cells, like peripheral mononuclear blood cells. Those do separate really nicely into T cells, B cells, NK cells, and I can actually identify them using a cell atlas. But for the colon, I just haven't been able to do that. So this is what I have to do. I just have to look at the expression across these figures here and then try to label these clusters here as a specific cell type. Again, this isn't finished just yet. I need to input a lot more details into this so that somebody can actually follow it. But for the most part, this is how we identify cell types in the colon.
So that's why, so you have potentially another approach that you were trying to optimize using the larger genes. You described it before a little bit. Is that something you're still working on, more general?
For which one? For this location of cell type?
Yeah. Was Victoria doing that? And you were taking I call it the personalized approach versus the more.
Yeah, so I know Victoria was working.
Oh, the cell states.
Yeah.
Right.
She's been working on that.
She's been doing some work in that. Still, yeah, I don't know the exact stat. She's been still working, but still classifying from all the clusters.
Okay.
Yeah, so what Michael presented, this is like a kind of way to put the biology knowledge into this annotation procedure. That's the correct way to do it. The other way, data driving or not, something knowledge transfer in the evolution that I don't trust. If you change the system, you try to make the name of the cell type transfer to that, it's not the way to go. This is the best thing.
Okay, so Michael, you're not wasting. Well, I never let wasting your time. But it's a highly labor intensive and requires huge amounts of prior knowledge. So Salim is going to probably never be able to do this because otherwise he would have to absorb all of this knowledge without having a biological experience. Michael spent years actually working in this field and through form of osmosis, active transport, and other techniques, he has assimilated this, which makes him extremely valuable to this process. And he has a good instinct too, also when he's generating garbage versus credible information. So, Michael, one thing that when you get through all this, I think the first question you want to ask is, are any of these treatments affecting the atlas? In other words, are the abundances of the population shifting? Because you have, again, a very unique biologic. He has a mouse that has no microflora in it. It was raised germ-free. And people do this quite a bit, but it's a completely artificial environment, and the animal's still alive and generally thriving relatively, but the biology is being massively impacted. Like you said, the immune system should be very immature, and the animal is clearly in a strange state.
Yeah, that's one reason why I started with this particular data set. It's more simpler.
That's
compared
simpler?
to- yeah.
Well, actually, so you have to figure out, can we show a control that has, so would that be just any mouse that we use Flex on, that we could say, okay, so here is a map from so it's not the perfect control for that notobiotic, but it is a control. In other words, it's the truth for normal colon. This is not normal.
Right.
It's very unique. It's not pathological. And then you can see whether the add-ons perturbed anything, maybe pushing it more normal,
Yeah.
without doing any real gene expression. We're just looking at something that, and the other thing is also just do a very quick lit search to find out how many papers are single cell in notobiotic mouse. It's got to be a very small list. But I'd just like to know, are you the first person to do this? I just can't believe it, but it is possible, which would make everything he's doing very novel, meaning that no one has ever created an atlas for a notobiotic mouse. And here he is. Michael's done it. And then he has manipulations where they added one bug and a substrate, or plus or minus a substrate for a bug. And maybe nothing changes from the atlas standpoint. And then you can move into any other complementary assay at whatever level, hopefully to answer maybe some of the questions our collaborators wanted. And that is a manuscript, really, because it's going to be doing something very interesting. Oh, they'll have the other information as well that could be added. So I think that this is wonderful. Anyway, it's good that, Salim, you see this, because we want you to be able at some point maybe